The last of the formed elements that we have yet to talk about are platelets. So that's the goal of this video. Once again, platelets occupy the region of blood we refer to as the buffy coat. Really, to be clear, there is no buffy coat in real circulating blood throughout the body. But once again, if we just put that blood in a centrifuge tube, spin it, sediment falls to the bottom. That sediment really are the red blood cells, known as hematocrit, which is roughly 45% of our blood. 55% of our blood is the blood plasma, and less than 1% is occupied by the white blood cells and platelets. Now, to be clear, blood is a fluid medium, and all of this stuff, all of these cells, red blood cells, white blood cells, and platelets, are flowing within the blood plasma. So it's not really separated in real time, if you will. What we're seeing over on the right of this image, all of those small green structures are to represent platelets. Platelets are extremely small, averaging about 3 micrometers in diameter. And to put that in perspective, large white blood cells like eosinophils and monocytes are roughly 13 micrometers, and the smallest of the leukocytes are about 6.5 micrometers. So once again, platelets at 3 micrometers in diameter are super small. A microscope is required to see these formed elements. Once again, platelets are formed elements because they are fragmentary cells. They are not true cells. They are formed via the process of thrombopoiesis. Hematopoiesis is production of all blood cells, all formed elements, erythrocytes, leukocytes, and platelets. Platelets derive from an extremely large cell known as a megakaryocyte, and that's what we see at the center of this image right here. A megakaryocyte spans in diameter roughly 150 micrometers, which can be seen with the naked eye without a microscope. Coming off of this megakaryocyte, we have these structures known as tendrils, and when those break off, they form into platelets, as we see over here. And to be clear, there are more platelets within the buffy coat, within the blood, than there are white blood cells. But platelets are so small that white blood cells still make up a greater volume of blood or a greater volume of the buffy coat than platelets do. To repeat that, there are more platelets than white blood cells. But because platelets are so minuscule, they occupy less volume than the white blood cells. Let's talk about some details on the platelets. Once again, they are anucleated. They lack a nucleus. Platelets are utilized to form what's known as a platelet plug. So they are involved in the process known as hemostasis. Hemostasis is the process that stops bleeding, or the cessation of bleeding. Anytime there's a laceration or cut in a blood vessel, whether that's external to the body or internal to the body, platelets play the integral role of stopping that bleed. And the first part of that is forming what's known as a platelet plug. In the process, platelets, and I'm looking at step 3 here, platelets are going to release a host of signaling molecules, sometimes referred to as factors. A factor is a moniker given to signaling molecules before it's really realized what they are. Are they a hormone? Are they a neurotransmitter? Are they a paracrine and autocrine? So don't worry about the details of the moniker factor, but platelets secrete signaling molecules that cause other events within the process of hemostasis to occur. For example, platelets are going to secrete serotonin, which is going to cause vasoconstriction, which is one of the first steps in hemostasis. And we'll talk about this with some images subsequently. Platelets are also going to release signaling molecules that convert inactive plasma proteins to active plasma proteins that help form that platelet plug. Platelets are also going to release signaling molecules that dissolve that platelet plug when we no longer need it. So the platelet plug is not something that's going to be in place for an indeterminate amount of time, and we'll discuss this. Platelets are also going to mobilize neutrophils and monocytes to the site of the laceration. And anytime there's a laceration in a bloodstream, there's the risk of bacteria entering our bloodstream. And because neutrophils and monocytes are phagocytic cells, they can help eradicate any bacteria, as can platelets, which we see with number 5 right here, where they can actually kill bacteria. So let's take a look at hemostasis. Once again, hemostasis is the cessation of a bleed, the stopping or eradication of a bleed in the body. And that process is going to involve first minimizing as much blood loss as possible, forming a plug, and then regrowing any tissue that was damaged. This is a blood vessel right here. In green is the lining of the blood vessel, which is simple squamous epithelial tissue, otherwise known as endothelium. The blue on the outside or bottom of the endothelium is connective tissue containing a large amount of collagen fibers. And then in red is going to represent the tunica media, or the smooth muscle of the blood vessel. I should have on the outside of the tunica media the connective tissue that makes up the tunica adventitia or tunica externa. I apologize for that. But once again, we have endothelium, connective tissue laden with collagen fibers, and then the smooth muscle making up the blood vessel. Okay, so the first thing that happens is going to be some sort of laceration or bleed from the blood vessel. That blood is leaving, passing through the layers of the endothelium, the connective tissue, and the smooth muscle. The first thing that is going to happen is platelets, which we do not see in this image. The platelets are going to start releasing serotonin. And to be clear, it's even possible that the endothelial cells of the blood vessel themselves are going to secrete serotonin. Serotonin is going to be secreted to cause vasoconstriction. So here we see vasoconstriction in the middle of the blood vessel. To be clear, if we look at the left side of this blood vessel, the very left side would be the heart, or at least the blood is coming from the heart. Blood in this case is flowing from left to right. So this site of vasoconstriction is upstream of the laceration. And the goal with the vasoconstriction, which is caused by the release of serotonin, serotonin is a signaling molecule. It also functions as a neurotransmitter in the brain and other parts of the nervous system. In this case, it's acting as a signaling molecule that causes vasoconstriction. The goal here with vasoconstriction upstream of the laceration is to decrease the volume of that blood vessel, which it does, to increase the pressure. Keep in mind, this is a chamber. The blood vessel is a chamber. By decreasing the volume, pressure is increased specifically in this area. Now, one may think that the increase in pressure is going to increase blood flow downstream or to the right of this vasoconstriction. And to be clear, it is going to immediately. But what I want you to think about is if you take a garden hose that is on, there's water flowing through it, and you put a crimp or bend in that garden hose, you're going to get an initial gush of water out of the garden hose, but then it's going to stop flowing from that point where you've crimped the hose. And if it doesn't stop flowing completely, it's certainly going to minimize dramatically. And that's the goal of vasoconstriction. It's the first step to help alleviate any great amount of blood loss from that laceration. Now, to be clear, we don't want total stoppage of blood flow through this point where we have caused vasoconstriction, because we still need other cells and molecules to come to the site of the laceration to help repair it and to help fight off any bacteria. For example, we talked about neutrophils and monocytes being mobilized to the area so they can ingest or phagocytize any bacteria that may arrive through the laceration. What we see here in this image are platelets in black starting to adhere to the broken wall of the blood vessel. Now, what they're really starting to adhere to are the collagen fibers of the connective tissue that underlie the endothelium. During normal times, when there is no hemorrhage, the endothelial cells release nitric oxide. Nitric oxide is a gas and has a number of functions in the human body, but with regards to blood flow in blood vessels, the nitric oxide prevents platelets from sticking or adhering to the endothelium. So in theory, platelets should never be able to stick to the endothelium, and that is a good thing because we don't want platelet plugs or any sort of clot being created in normal times where we need blood to continue flowing to perfuse the body with oxygen and nutrients. But if the collagen fibers of that underlying connective tissue are exposed, then the nitric oxide plays no part, one, because there's no endothelium in this particular space, which we see right here, and then the platelets start binding to the collagen fibers and start binding to each other. And those platelets are going to start releasing PAF, which is known as platelet activating factor, which is going to attract more platelets to the site of the laceration. And this is what's known as positive feedback. I will go into positive feedback in more detail in a physiology course, but in short, it's causing more platelets to come to the site, which those platelets release more PAF, platelet activating factor, to attract more platelets, and more platelets come to the area. And that's what we see in this image here, just a greater aggregation of platelets at the site of the laceration. And then to really finalize that platelet plug, we need fibrin. Fibrin is a sticky protein that causes those platelets to all form a great sticky mass to prevent any more loss of fluid from that blood vessel. Fibrin is a plasma protein, but it is not always flowing through the plasma. As a matter of fact, it never is until fibrinogen, the inactive form of fibrin, is converted to fibrin. So I will say that once again. In our blood, we have a number of plasma proteins that we've spoken about, such as the globulins, albumins, and fibrinogen. Fibrinogen is an inactive plasma protein. It doesn't really do anything in the blood with regards to hemostasis unless there is a laceration. And when there is a laceration, the platelets are going to secrete a signaling molecule known as thrombin. Thrombin will convert fibrinogen to fibrin during times of a hemorrhage or a blood loss. Then that fibrin is going to ensure that that platelet plug is adhering to each other. The platelets are adhering to each other and adhere to the remaining wall of that blood vessel to prevent any more blood loss. So the combination of the upstream vasoconstriction and the platelet plug with the fibrin is going to be enough to prevent any more blood loss. So at this point, the cessation of the bleed has happened. That is to say, we no longer have any more blood loss. But we don't want that platelet plug to remain there indefinitely. Because if nothing else, this has actually decreased the lumen of the blood vessel. And by decreasing the lumen of the blood vessel, you're increasing the pressure here, minimizing blood flow downstream. And we don't want that indefinitely. So the next step is going to be repairing the tissue wall of this blood vessel. And once that tissue wall is fully repaired, then dissolution of the clot can happen. So here we have the vessel wall has been repaired, and that's achieved by the secretion of growth factors from the platelets. And then once it's fully repaired, plasmin comes in to dissolve the clot. Plasmin is a plasma protein that is not always present, but its precursor, plasminogen, is certainly present. Plasminogen is another inactive plasma protein that's continually flowing through the blood and only gets activated once the vessel wall has been fully repaired. Once it's fully repaired, the platelets are going to release a tissue platelet activator, which converts plasminogen to plasmin. Once again, plasmin is going to dissolve that clot. And that is the Cliff Notes version of hemostasis and the role of platelets within the cardiovascular system.